Svante Paabo got his PhD from the University of Uppsala. Or was it Uppsala?
Yeah, Uppsala.
Uppsala in Sweden, and did 4 years postdoc at Berkeley. So he's been in the Bay Area before. He's actually the founder of the field of ancient DNA, who even when he was a postdoc, or when a doctoral student rather, was working on sequencing DNA from Egyptian mummies without telling his advisor in his spare time. But fortunately, it worked out well. And much later, anytime you read about Neanderthals and whether they mated with humans, where they came from, chances are it came from his lab, the Max Planck Institute in Leipzig, Germany. So please welcome Svante Paabo.
Okay, so do that afterwards if you think it's So, yeah, what I wanted to do though was then talk a bit about what we can learn about the origins of modern humans, so sort of the origins of the type of humans that exist all over the planet today, from studying the genome of our closest extinct relative, the Neanderthals. And the technology and the ability to do this have come about over 30 years of sort of development, and the sort of history of that happened is sort of what's described in this book that just appeared. It sort of discusses not only the science, but a bit how this came about, including the conflicts and the mistakes I made on the way and things like that. But what I don't want to recount here exactly what's in the book. What I'd rather do is sort of talk a bit about the insights that has emerged over the years about human origins from this work. And in the end, a bit talk about where we want to go in the future in the next 5 or 10 years or so. But just before we start, let me then remind you about what you all know, I think, that our genome, the DNA, is stored in almost all cells in our bodies on these chromosomes in the form of the famous DNA, this double helix. As you also know, the information is there in the sequence, in the form of the sequence of these 4 bases, A, T, G's, and whatever I forgot, A's. And it's there twice, so to say, and in a complementary form. So whenever there's an A on one strand, there's a T on the other. When there's a G there, there's a C there. And that's really important, because whenever a cell divides, particularly then in the germline, where new individuals may be formed, there are enzymes that unwind those strands, and 2 new strands are synthesized with the old as a template. So you get 2 fairly exact copies, again, of the genome. But nothing is, of course, perfect in nature. So sometimes an error may be made, and if that error is not repaired quickly enough before the cell divides again, that will appear then as a mutation, a new version of a DNA sequence. So any baby that's born has in the order of 100, 200 new mutations that's there neither in the father nor in the mother. And we can detect the effects of those mutations in the population. If we compare DNA sequences of 2 people in a room, we will find a difference every 1,200, 1,300 bases between us. If we add in a chimp, we will find a difference every 100 bases or so. So if you want to reconstruct the history of a piece of DNA, what you do is use our best models for how these mutations happen, and reconstruct the history and depict it in the form of a tree like this normally, where in this case it's super simple. The 2 human sequences go back to a common ancestor rather recently, and much further back is a common ancestor shared with the chimpanzees also. And as you also know, our genome is a bit over 3 billion base pairs. So if we compare 2 genomes, either the 2 genomes you carry from your 2 parents or 2 people in the room, we have in the order of 3 million differences between 2 individuals. So there's quite a lot of information there to sort of reconstruct the history with. And if you do this on a global scale, what you will find is that in Africa, you have more variation than outside Africa. So although there are only 700 million people or something like that living in Africa, and almost 7 billion people living outside Africa, we have more genetic variation among those Africans than among all the non-Africans taken as a group. And not only that, most of the DNA variants we find outside Africa, we find closely related forms also inside Africa. But in addition then, there is a component of the variation in Africa that's not outside. So the interpretation of that is then that modern humans evolved, emerged in Africa, accumulated variation there, and a part of that variation went out and colonized the rest of the world. And by genetic tricks, you can also figure out approximately when this happened by looking at how big chunks over how big distances are things associated on a chromosome and things like that. And you find that that's fairly recently. In the order of 100,000 years or even a bit less, do people start expanding out of Africa. So this is the origin of sort of this recent African origin model of modern humans. But there is, of course, a problem with that, and that is that we know from finding bones that there were other forms of humans around since a long time, since 2 million years at that time. So most famously, there were Neanderthals in Europe and Western Asia. In Eastern Asia, there were other forms of now extinct humans that are less well described. So a big question is sort of what happened with them, how are we related to them, did they contribute to us? So, but to study their DNA, you would have to go back in the bones, of course. And that, of course, is a bit difficult. And this then goes back to sort of work that started in the 80s, where I had studied Egyptology, actually, and then ended up in graduate school studying molecular genetics. So I knew there were thousands and thousands of mummies of animals and humans around that look very well preserved. I thought perhaps there's DNA preserved in them. And started looking at in the microscope on tissues from such remains. And that turned out to be very frustrating. So this is a muscle from a present-day person. You see the muscle fibers. You see these black dots, which are the cell nuclei where the genome is stored. And this is a muscle from a 2,000-year-old mummy, much like the one we looked at there. So you barely see there are muscle fibers there. You see no cell nuclei. There's no DNA preserved. But it's sort of not always that depressing to look at these things. So it's also described in a book of how at that time in the 80s, lived in Sweden, so you had rather good connections to communist countries. You were able to get mummies from East Berlin. And one of the mummies there, this 2,400-year-old child, you could, in the sort of basal levels of the skin, see things that looked like cell nuclei. You could actually stain it and show DNA is there, and go on to extract the DNA, replicate it in bacteria, and show that some of it came from a human. So in hindsight, I really believe this microscopy, that the cell nuclei here contain DNA. That's for sure DNA from that mummy. The DNA I replicated and published is for sure a contamination. It's for sure DNA from me or the museum keeper in East Berlin or something like that. Because what has since become clear is that there's generally very little DNA preserved in such remains. It's degraded and chemically modified. So whereas, say, from a gram of tissue, you might extract a microgram of DNA from a blood sample or from present-day tissue, there's something like 10,000-fold to a million-fold less in such old remains. And the vast majority of the DNA here comes from bacteria and things that have grown in the tissues since it died. So if you have tiny amounts of contamination from present-day humans here, so say skin fragments in a room like this where people are around, sort of dust particles are to a big extent skin fragments that do contain a lot of DNA. So if such comes into a present-day experiment, it will be swamped out by the billions of copies of the modern DNA there. But in these ancient experiments, if a dust particle lands, it can easily overwhelm the whole experiment. So it's also described there how we sort of over the years become more and more paranoid about this until when we today now work in sort of spacesuit-like things in sort of rooms with high pressure and filtered air so dust doesn't get in, pretty much like a chip factory or something like that, to avoid contaminating the experiments. It also focused a lot on not the nuclear genome that comes from the moms and dads, but a tiny part of the genome outside the cell nuclei that comes only from the mother, the mitochondrial genome, because there are many, many copies of it. per cell, so bigger chance that a little bit survives. And we worked ourselves back in time. So it started when I was here at Berkeley. Actually zoological collections at the museum at the university there of these kangaroo rats from the Mojave Desert that had been collected 70, 80 years ago. So we went out and set traps with the field maps from that time and collected rats now and could follow sort of the populations then over 70, 80 years. To a bit older extinct animals, marsupial wolves, say, that became extinct about 100 years ago from overhunting in Australia, or things that disappeared in the last glaciation. So these giant ground sloths, for example, in North and South America, or mammoths and mastodons and things like that. But our big interest was really Neanderthals as our closest relatives. And so Neanderthals appear 300,000-400,000 years ago, and then exist in Europe and Western Asia until they become extinct something like 30,000 years ago in connection with that present-day humans appear. So a big question was then what happened when modern humans encountered these guys? And there are two sort of ideas about this that one was fighting about in paleontology, vicious fights that paleontologists can have over 30 years, where one idea is modern humans come out of Africa, meet these guys in Europe and Asia and replace them with no mixture whatsoever, that there's no contribution from Neanderthals to Europeans today, for example. And the other idea is that when modern humans come to these regions, they do eventually replace these forms, but they do mix with them, so that Neanderthals would have contributed genetically to present-day Europeans, and these other forms in Asia to present-day Asians. So you can see this as a sort of sliding scale from total replacement, 0% contribution from these earlier guys, to sliding scale to more and more of that. So our first chance to sort of test this came in 96, when after a lot of intrigues, we got access to this specimen, which is a Neanderthal, but it's not just any Neanderthal. It's the Neanderthal that was found in 1856 in Neander Valley and gave its name to this group of humans. And we were really lucky, I think, that the first Neanderthal we studied was really the type specimen that gave its name to these guys. Because whatever Neanderthal we since have studied, there's always some paleontologist to come and say, it's a little too gracile, it's a little too robust, it's not quite typical. But if this is not a Neanderthal, then Neanderthals don't exist, so to say. So with the technology at the time, we sort of retrieved a part of that mitochondrial genome with great effort, short little pieces, many, many times over, looking for the things that were consistently there. And then reconstructed such a phylogenetic tree for the mitochondrial DNA, then, this tiny part of the genome. And what we saw was that what was already known, that all mitochondrial DNA of everybody living today go back 100, 200,000 years to a common ancestor of all the mitochondria that exist today. But the Neanderthal mitochondrial DNA go much further back to common ancestor, 500,000 years or so. So they look quite different in their mitochondrial genome. So in this sliding scale of things, it's really here. There are no humans today that run around with a Neanderthal mitochondrial genome. It's total replacement, no contribution. So that was then the picture for this mitochondrial genome, and we couldn't get to the nuclear genome. And I think I'm on the published record 8 years ago or something like that of saying we will never see the nuclear genome of Neanderthals. And you should, of course, never make negative predictions like that, because you're generally overtaken by history and by technology, actually. And in this case, what I didn't anticipate was really high-throughput DNA sequencing. That came around in the early 2000s, the ability to sequence millions of DNA molecules really rapidly and inexpensively. So instead of trying to find some little piece you're particularly interested in, you could simply extract all the DNA from such a fossil, sequence all the molecules you find in there, make your little data bank, and start comparing it to the human genome that had then become sequenced and say, what has similarity to the human genome that presumably comes from the Neanderthal? So the first place where this worked was from a cave site in southern Europe, in Croatia, this place, from this bone that's 38,000 years old, from this little fragment of the bone here. And the first thing you then find is that the fragments of DNA are really small. So whereas from a blood sample from me, you would extract things that are 10, 20,000 base pairs long, here is really 50, 60 base pairs, tiny little fragments. And the vast majority of the DNA doesn't come from the Neanderthal at all, but from bacteria and fungi that colonized the bone when it was lying there in the sediments in the cave. So our very best Neanderthals have something like 3%, 3.5% endogenous DNA. So we then managed to raise some money to, over 5 years, work a lot on the technology from which we can extract the DNA from the bone and manipulate it into a form that you can feed into the sequencing machines. And we have gotten a lot more efficient than that, something like 500-fold more efficient over this time. And the machines have also gotten better in how much they can sequence. We looked through a lot of different Neanderthal sites and bones to find the ones with the most DNA in them. And it sort of describes lots of little problems in running a group with different forms of expertise to actually get this done. We identified then 3 bones that were particularly good that we used for this, and sequenced a bit over 1 billion DNA fragments from these bones, which at the time was quite a lot. Vast majority, of course, not from the Neanderthal, as I said, but from bacteria. But we could then match the ones that had similarity to the human genome with computer programs we developed to the human genome, taking various types of errors that occur in these sequences and so on into account. And we ended up with a sort of first view of the Neanderthal genome in 2010 then, where we had seen a little over half of the genome, at least once, from these fragments. So we could begin asking questions. And that then involved a lot of sort of population. Yes, please.
With fragments just being 20 bases, are they unique enough to figure out whether something is the same gene, or
yes.
Could the Neanderthals have had genes which are completely different from humans, but which we learned?
Yeah. Well, so first of all, if Neanderthals had something that at all doesn't exist in the human genome today, we would be totally blind to it. But that is probably very little such material there. If you compare to the apes, for example, there's hardly any such things. But a big, big challenge is this matching of these fragments. Because, yes, we sort of have a cutoff of 30. We don't regard anything below 30. But also there is a sort of an issue about we have a vast majority of bacterial DNA that can have spurious similarity to places in the human genome. We certainly mismap some things, but yes. So that is sort of one of the big things is actually an informatics problem here. But so we then involved a lot of more population genetics, modeling people. Particularly, I should mention Multislacking at his group at UC Berkeley, and David Reich and Nick Patterson at the Broad Institute in Boston, to sort of analyze these genomes. And one of the first questions was, of course, what happened when modern humans came out of Africa and met Neanderthals? Did one mix or not? And we addressed that in many different ways. But one way, the most intuitive one, is to say, well, if there was a contribution from Neanderthals to present-day people in Europe, we would, of course, expect Europeans today to share more genetic variants with Neanderthals than people in Africa, where there have never been Neanderthals, so they couldn't have contributed there. So this is this idea again, right? Saying if there is no mixture whatsoever, the Neanderthal is just as far from people in Africa as people in Europe. If there is a contribution, on average, Neanderthals are more similar to Europeans than to people in Africa. So we then had the Neanderthal genome, and we needed to compare to present-day genomes and look for small signals They were, of course, very scared of sort of errors in the sequencing or differences in error profiles in different genomes. So we went out and sequenced 5 genomes totally in parallel from present-day people ourselves to know we had the same error profiles in them all. So wanted one European person. To us, of course, the archetypical European person is someone from France. So we sequenced a French person. We sequenced 2 people from Africa, one from China and one from Papua New Guinea. And then we did a very simple analysis. So just to test this first, we compare the 2 present-day people, 2 Africans, and find all the places where they differ in their genome. And then we just say for the Neanderthal, how often does it match one African and the other African? Since Neanderthals have never been in Africa, there is no reason for a Neanderthal to be more close to one African than the other African. It should be equally distant, right? And indeed, statistically speaking, it's 50-50, say 99,000 or 100,000 matches there. When we do this with the European person and the African person, then we found, to my surprise actually, statistically significantly more matching to the European than the African individual. Even more surprising was that in China, relative to Africa, we again find it, and in Papua New Guinea, again. So this really surprised me. I was biased after the mitochondrial genome saying there had been no contribution, but if there was one, I would of course expect it where Neanderthals had existed in Europe. But we found it in China, where most people agree Neanderthals have never been, and in Papua New Guinea, where everyone agrees Neanderthals have never been. So the model we then ended up suggesting that since have been borne out by other work is to suggest that when modern humans came out of Africa, they presumably passed by the Middle East. And we know that in the Middle East there were Neanderthals. So if these early modern humans mixed with Neanderthals and then became the ancestors of everybody outside Africa, they can sort of carry with them out into the world this Neanderthal contribution, also to regions where Neanderthals have never existed, to the extent that 1 to 2% of the genome, of the DNA of people in these parts of the world come from Neanderthals today. Why didn't we find any mitochondrial contribution when we now see it in the nuclear genome? I think the simplest, boring answer is that it can be simply by chance. The mitochondrial genome is inherited as one unit, and it can just be lost stochastically. But it's of course also true that the mitochondrial genome is maternally inherited. So we get it from our mothers and not from the fathers. So if it was particularly Neanderthal men who contributed to the modern human gene pool, we would also bias against mitochondrial DNA. And of course you can say even by normal consideration saying if you have hybrids, you would of course expect the babies to stay with their mothers, right? So one would perhaps, and we know that these individuals stayed in the modern human population because they in turn had kids and contributed to people today. So maybe we're particularly picking up the cases where the mother was modern human and the father was a Neanderthal. But sort of both are possible. Yes, so we're sort of blind to contribution in the other direction into Neanderthals simply because we would then have to find a Neanderthal that lived at the right place and late enough in time that its ancestors had met the modern humans. So in reality, we have only something like 300 Neanderthal individuals or so distributed over hundreds of thousands of years of history. So we would be extremely lucky to find someone like that, I think. So we haven't done that, and perhaps we will never. Maybe, in the Middle East or something like that. Can we differentiate against a scenario where just modern humans and Neanderthals have a more recent common ancestor than we think? And I think really can. I mean, we see this difference between Africa and non-Africa, right? We see that overall in the genome we are all closely related, including all people in Africa, of course. But people outside Africa have this extra component in their genome, a tiny little part that is deeper diverged, yet very similar to the Neanderthal genome. So that's really the only reasonable explanation for that is from Neanderthals. So the question is, the offspring must have been fertile, and can we estimate sort of how fertile they were? Or so the only thing we can say about that's sort of very recent work now, where two groups, one project where we were involved together with people at Broad and another group in Seattle, have independently now designed pretty efficient algorithm to go over present-day human genomes and say which parts actually come from Neanderthals. And if you do that in thousands of genomes of present-day people, you will find regions nowadays where we see no Neanderthal contribution, although we would expect it from the distribution across the whole genome, but then there are what we call deserts, where there suddenly is no Neanderthal contribution. So that's interesting, because you could imagine they are hiding something that really defines us as modern humans, that's selected against when it comes from Neanderthals. And if we then look at what genes are in such regions and where are they expressed in our body, the only significant sort of overexpression of these genes are in the male germline, in testicles. So it really suggests that there may have been some problem in the hybrids with male fertility. And that's actually a rather common thing when sort of closely related species or groups mix, that the male hybrids have problems with fertility. So horses and donkeys, mules, right? The males are infertile, but the females are fertile. So it seems to have gone on something like that. It may have been that the boys didn't do well in reproduction, at least. But that's really the only thing we can at the moment say about that. But there seems to have been some incompatibility there biologically, really. So I can never sort of stop myself from sort of also pointing out that there have been a lot of scientific interest in this, lots of follow-up things, but the public is also very interested. And lots of people write to us, and a lot of people write and self-identify as Neanderthals and or volunteer to give us blood samples and things. And after a while reading these mails, I started seeing a pattern. And that was that it's mainly men who write to us and self-identify, and very few women who write and say they are Neanderthals. And so I sort of presented this as my research to my group, counting emails, you know. And they said, it's just ascertainment. It's just because men are more interested in molecular genetics, that they will write to you, women will not write to you. But that's not true, because I went back and looked at the emails again, and there are plenty of women who write and say they are married to Neanderthals, actually. Whereas there's so far not a single man who have claimed he's married to a Neanderthal woman. We're very interested in other forms of humans also. So there are lots of other forms of Neanderthals one finds that one can hope to study now. And we work together with Russian colleagues that excavate in Siberia, and particularly then in this cave, the Niseva cave, in southern Russia on the border to China and Mongolia. And it's a very beautiful area, and this cave, where they in 2008 found a tiny little bone that they were actually extremely skilled in realizing that it might be a human bone. So it's a fragment of the last phalanx of a pinky from a little girl. So we got this bone and we extracted DNA from it, and were surprised to find that it was very much endogenous DNA, like 70%, very different from the Neanderthal samples, the best Neanderthal samples. Meaning really coming from this individual. And we don't know why it's so well preserved. It's not permafrost here, it's not frozen. But we were able to sequence a genome from this girl too. And we're quite surprised to see that it was not a Neanderthal and not a modern human. It has a common origin with Neanderthals, but far back, 200, 250,000 years back. And since then, Neanderthals have a long independent history. So this is the first time actually that only based on a genome sequence you define a sort of new group of extinct humans that we then called Denisovans, after this Denisova cave where they are found, just like Neanderthals are named after the first place where they were found. And since then we have worked on new techniques to be even more efficient in extracting tiny amounts of DNA. So we now have a very good genome from this girl. We've sequenced every position many times over in the genome. It's actually better quality than most genome sequenced from people today. So we can begin to see, when we have so good genomes, see cool things. We can, for example, see missing mutations here. This person lived tens of thousands of years ago, so compared to us today, they have experienced fewer mutations, and we can actually measure that. So relative back to the common ancestor with the chimp, there's a little over 1% of the mutations that are missing here. So if we now just say that's 6.5 million years ago, we can actually date the bone to approximately 60 to 80,000 years ago. So it's sort of fascinating to me that from a bone that's too small to do a carbon date, when we can sequence the genome, we can actually now begin to date it from the genome. There are still caveats about this, particularly error rates in the present-day genomes confuse this number, but it's a sort of certainly something that will come in the future. We can ask us for the Neanderthals, as for Neanderthals, did they contribute this Denisovan guy to present-day people? And yes, they did, in Oceania, not in Siberia. So this then suggests that they were more widespread in the past. I think that they have been also here meeting ancestors in that area. So I think it's sort of an indication of what will come in the future. This is a copy of this tiny little bone, where we can then get a whole genome sequence, we can date it, we can reconstruct a lot about the history of this group, but we don't know how their skeletons look. We don't know what stone tools they made. And I think it's something we'll see a lot more in the archaeology in the future. Yes. So it shouldn't be too long. We have from this cave also then found a little toe bone that turned out to be Neanderthal. So we now have a very good Neanderthal genome also that we've sequenced many times over to very high quality. One of the cool things we find then is that you can, of course, then walk over the chromosomes and find where there are differences between the two genomes that this person inherits from their mother and their father. And what we then find in this Neanderthal is that we find huge segments. This is 19 million bases here where the two chromosomes are identical. And we found much less of those in this Denisovan or in present-day people. And this, of course, means that the parents of this individual were very closely related. So we can sort of reconstruct the relationships that must have been, one of these four relationships must have been for this Neanderthal individual. The parents must have been half-sibs, or say grandfather, granddaughter. Don't even ask me what double first cousins are. I can never reconstruct it. But they were clearly closely related. So it will be very interesting to see if that's a general pattern in Neanderthals when we get more genomes in the future. So we have now a bit more things there. We can actually see how these things have mixed with each other. Then Neanderthals to modern humans, Denisovans to modern humans. We can see on the mainland Asia also a bit of a contribution from Denisovans. And we can see, quite interesting, a contribution from something else, a deeper divergence into Denisovans, not into Neanderthals. Probably Homo erectus or so in Asia. So let me accelerate a little bit. I'll not do this. I want to just in the end come to where we go now. So just to wrap that up, we have sort of excluded this absolute no contribution, total replacement. But we see a bit of replacement from these earlier forms to present-day humans. But the big picture is, of course, still replacement coming out of Africa. So sort of as a term for this, I sort of suggest leaky replacement or so. The big picture is replacement. There's a bit of contribution from these earlier forms. And what can we now do with these genomes? I think when we now have high quality genomes from our closest relatives, the really cool thing is we can focus on these genetic changes here. Things that we all have in common on the planet today, but where the Neanderthals and Denisovans look like the apes. So things that appeared in modern humans and spread to everybody in the last little segment of our history. So that's sort of, why is that particularly interesting? Well, I think there are a number of things that changed with modern humans. A big thing is technology, of course. Neanderthals appeared 350,000 years ago, disappeared 30,000 years ago. The stone tools they make in the beginning of their history and the end of the history are pretty much identical. Modern humans appear, say, 100,000 years ago, and I think we agree, particularly at this place, for example, I think that technology today is different from 100,000 years ago. So technology starts changing rapidly with modern humans and can become regionalized, for example. Neanderthals made the same stone tools wherever they were, from Central Asia to Western Europe. With modern humans, it changes so rapidly, so when you don't have much communication, technology really becomes being different in different regions. Art that really depicts things comes only with modern humans. And modern humans, of course, start spreading across the whole globe. These other forms of humans had lived in Africa and Eurasia for 2 million years. They never made it to Americas, never to Australia, never to Madagascar in 2 million years. Modern humans start spreading out of the Middle East 60,000 years ago. And those 60,000 years come to every speck of land, right? Not only the big continent, but every little island out in the Pacific. So there's actually no evidence that Neanderthals or the other early humans ever spread across water where you don't see land on the other side. So they were not mad. But modern humans, how many people must not sail out on the Pacific before you found Easter Island and just perish? And we can never stop right now. We go to the moon and we go to Mars. So there is some craziness there also, not only technology, I think. So we can look in the genome now for all the things that we have in common that is not shared with the Neanderthals. So if we take the strict approach of saying things to our best of our knowledge, 100% of humans have today, but Neanderthals and apes don't have it, then that's not a long list of things. It's surprisingly short. It's something like 30,000 changes in the genome. So whereas I differ from an individual in this room by 3 million differences or so, if we say things that we all have in common and the Neanderthals are different, it's just 30,000. So you can scroll through them in 2 hours and look at them, actually, in the computer. So, for example, there's just 96 changes in proteins of amino acids in the... So this is a list of all the genes that have such differences. The dirty little secret of genomics is, of course, that we are incredibly bad at predicting what such changes would have for consequences. But we're, of course, particularly interested in things expressed in the brain. And we and many others will now start to look at that. So just to give you a feel for this, if we just look in different layers of the brain during development in the fetus, the one area within the red lineage there, we see a significant overrepresentation of those proteins that have differences to the Neanderthals is in the proliferative zone, where the neuronal precursor cells divide to make new neurons for our brain. And actually 3 of those 87 proteins are expressed in the spindle and the part of the cell that pull the chromosomes apart in cell division. And it's known that the plane, how these cleavages take place here, determine what type of neurons will be formed during brain development. So these 3 genes, for example, are particularly interesting to study, and I think we and many others will now start doing that. So just to end then, what we then want to study is human-specific changes. So how will we do that? Because it's, of course, a problem. We have no animal model, really. So there are people like George Church at Harvard who have suggested we should clone Neanderthals. We should engineer these changes into modern human stem cell and have babies born that are essentially Neanderthals. And I think there are many reasons to say it's actually both technically not feasible in my mind, but leaving that apart is sort of ethically, of course, totally unthinkable. But what can we do? I think something that will come is, of course, the genome is a small place after all. 3 billion bases. I already told you that there are 200 new mutations in each baby that is born. There are 7 billion people. So every position in the genome that can change and is compatible with human life exists out there in the population, but generally at low frequency. But in a future where we will all have our genome sequenced, when we just walk into the doctor's office, so we'll have millions and millions of DNA sequences, we'll be able to start find people who have back mutations to the ancestral state and study them. So that will become possible. You can, of course, also engineer in these changes into stem cells and differentiate tissues, nerve cells, liver cells in test tube in the lab, and we and others starting to do that. And you can start putting these things into mice and start humanizing parts of the mouse brain, say, or something like that. So just to end up, I think that I have convinced you that if you're interested in what makes humans unique, particularly this last part of human history that set us on this very unique trajectory of spreading across the whole globe, coming to dominate even parts of the biosphere today, then it's very useful to now have the genomes of our very closest relative, the Neanderthals, but to now go on to understand those changes. It will not be enough to just stare at the genomes. We have to do functional work. And I think one of the most realistic ones is actually this thing to humanize mice. So that was this. Glad to answer more questions.
How much of a sample do you use up, and how much do you leave for future technology?
Yes. So to just test if there is DNA preserved that one could study, we use like 50, 100 milligrams, so very, very little, actually. But then if there is DNA there and one wants to go on, then it really depends on the preservation state. If it's very little DNA, we need more. But we never work with more than half a gram or maybe up to a gram of bone material. So it's not that we use up enormous amounts.
Excuse me.
Yes.
How many individual Neanderthal genomes do we have sequenced?
So we have only one really good quality one. We have then this low quality one from Croatia and one from the Caucasus that I didn't talk about. So we have 3 genome sequences. We have some exome sequences too. We just have the protein coding parts on 2 more individuals.
Could you use this to create technology for archaeologists like fish stains or something to help them find more Neanderthal DNA?
Yes, so what we can do is to take small samples and determine the species of bones where you don't know what it is. And we begin to do that, the vast majority, of course, animals, but now and again we then find new Neanderthal bones. And then you're in a good situation, because then you can really argue, if you didn't even know what species it is, it can't be very valuable to you, this bone. You should give it to me. So.
Yes, have there been any specific interesting insights about the X or Y chromosomes of Neanderthals compared to ours?
Well, so these sort of have, very curiously, these are female individuals so far we have happened to hit in those 3 individuals. But so on the X chromosome we see very little contribution from Neanderthals. It's probably again to present-day people. So it's almost like a desert, the whole chromosome, which is probably also compatible with that the men had particular problems. Of course, there are extra many genes on the X that has to do with reproduction. And males are, of course, slaves to their X chromosome, if you like. Men have only 1 X chromosome, so if they get bad alleles, they are in trouble, whereas females have 2 chances also on the X. So it's probably, again, a reflection of that the men may have had a problem in the hybrids. But that's the only thing at the moment we can say about that, I think.
The contaminant DNA that you found in the samples, was that contemporaneous with the death of the individual?
The bacteria that are there. We don't really know. We can see there are typical chemical modifications of the old DNA. And in some specimens, most of the bacteria have such things. So they're at least thousands of years old. But we don't know how much of that goes back to, really 60,000 years ago, and how much can be.
Presumably, you couldn't look at the gut microbiota because that would have been completely eroded away, not in the skeleton?
I would think so, yes. But yes, it's an interesting question, and we've talked about studying the bacteria from the soil just outside the bone and inside the bone and comparing that and see if one can fish out something. But
So since the DNA or the bone fragments were found in Europe, how do you know that the contamination is not European, from European humans?
yeah,
How do you control for that?
That the admixture signal is yes. So I mean, there are a lot of more analysis than what I present here that sort of really indicate that. For example, if you look in the present-day humans, in the fragments that are particularly close to the Neanderthal, say in an individual, in me, those fragments are also particularly different from everyone else in Europe. So if you just plot, say, fragments where I'm closer and closer to a Neanderthal, most of that is simply due to that these are regions of the genome that change slowly. But then I also get closer and closer to other Europeans today. These are just regions that change slowly. But when I get really close to the Neanderthal, then suddenly I'm more different from everyone else. So that's another line of evidence to say that these are really things from the Neanderthal.
Let's thank him for coming here.